Hello, I'm Sarah Ann Harris, and I'm a computational biophysicist from the School of Physics and Astronomy and the Asprey Centre for Structural and Molecular Biology at the University of Leeds in the UK. So, what I would like to tell you about is why I think that the future of biology is biophysics, so bio to the power of physics, but squared. And it's squared because there are two distinct ways in which physics can make a contribution to the biological sciences. So this futuristic statue here, for me, summarizes the big burning question in the biological sciences, which is how to go from the genotype as encoded by the DNA through to the phenotype. So how do we know from the sequence of the DNA what the phenotype of the organism is going to be? And I would like to describe how I think physics can help us on this journey. So just a quick tour of the physics that we understand. So one of the really inspiring things about physics is that it covers everything from the very, very tiny, as we can see with things like particle physics experiments, so the Large Hadron Collider, all the way up to literally being able to think about the whole universe and what it contains. And one of the big powerful aspects of this is summarized by the electricity and magnetism at the top. So we understand electricity and magnetism so well that we've actually got 4 equations that enable us to calculate pretty much anything involving electricity and magnetism that we would like to know. So if we can just work these things out with a pencil, or most normally with a computer, but we can just calculate what the magnetic or electric field in a particular situation should be using these equations, Maxwell's equations. And we can understand, we can engineer materials, we understand quantum mechanics, in fact, to such an extent that I've even got quantum mechanics on my washing powder. And of course, this has been absolutely, it's completely changed the world. So we have nuclear power. This is a picture of a steam-powered fire engine from Leeds, and of course it was our understanding of thermodynamics, of the relationship between heat, energy, and work that enabled James Watt to invent the first steam engine, and it drove the start of the Industrial Revolution actually just across the hills in Manchester in the UK. And of course, it's also our understanding of physics that has enabled us to generate semiconductors and to explore even down to very, 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 very tiny temperatures. So this very powerful toolkit has got 2 potential extremely important applications also in biology. So firstly, the tools of physics can help us design new experiments and new experimental technologies. So, I mean, arguably the most well established is X-ray crystallography and, of course, NMR. Both of these are originally physics techniques being incredibly successfully applied in biology. We're now undergoing the resolution revolution in cryo-EM and cryo-electron tomography, and this is me and Hua Chu at Stanford in front of his wonderful new sets of cryo-EMs. And the technology that drove the improvements in cryo-EM and cryo-ET is the detector technology, and of course that's done by physics and engineering. Now, I've just listed just a few tools here that have also come out of physics, primarily microscopies. So here we have a super resolution microscopy image actually taken from the axoneme. And things like super resolution, light sheets, DNA paint, all of these optical technologies are, of course, tools that originally come from physics. Similarly, the single molecule technique. So we have an example here of a DNA mini circle imaged by AFM. There are also optical and magnetic tweezers that we can use to pull and stretch molecules and investigate their mechanical properties. So physics can give us new biophysical tools that can help us see and measure things that we couldn't do previously, and that's extremely important. The other thing that physics can potentially give us from the other side is new understanding. So if we can write down a set of equations that describe the physical and chemical interactions within our system to sufficient accuracy, then in principle, we can predict what it's going to do. And this is, again, me. And this is at San Diego Supercomputing Center. And these sorts of supercomputing technologies can be used to run simulations such as the one on the right-hand side, which is an atomistic molecular dynamics simulation of actually this DNA mini circle, the one on the left-hand side. And these use bespoke physics equations. So we put all of the physics in and evolve the system as a function of time. And this gives us what has been termed the computational microscope. So there's this idea that the computations allow us to see things that we couldn't see with experiment alone. It's like having a microscope so powerful in this instance that we can see the actual atoms within the system. So this picture at the top of me laying flowers on the grave of Boltzmann, this is to remind me to say that, of course, also a lot of the tools we use to refine structures, such as cryo-EM structures, actually originally these sort of maximum likelihood methods for ascribing uncertainty, a lot of those originally come from statistical physics, of which, of course, Boltzmann is the father. So the two types of progress, the two types of help we can get from physics in biology, we can get new experimental tools. And when we've made the measurements with those new experimental tools that we've got from our physics, we can then build those into a predictive model using equations that we then hope gives us more insight and understanding of the system. So if we focus for a moment on DNA, omics, genomics, DNA biophysics, then there are two distinct ways of thinking about DNA in this context. So we can either think about DNA in what I would call the bioinformatics type way. And in this view of DNA, it's very abstract. We just treat the DNA as a 4-letter chemical code that could be read like it's in a book. We could write it down and read it like it's in a book. And this is, of course, the sort of information that we get from sequencing. So this is one way to think about DNA. But the other way to think about DNA is we can also consider it as not just a chemical code, but as an actual physical object that has a shape. It has interactions with other molecules due to its chemistry. It has a flexibility, etc., etc., etc. And of course, if we think about DNA in this way, it's more complex, but of course it contains more information. And the important thing is that if we're going to understand how to go from the genotype to the phenotype, we need to not just know what DNA is there, we need to understand how it is read, how it is regulated, and how it is processed. And we can only understand that if we think about it in terms of a physical object and how it interacts with everything else. We're not going to be able to understand the full complexity of the situation if we just treat it as a four-letter code. And for an interesting philosophical discussion of the difference between deductive and inductive science, then I recommend this nice paper in the Journal of the Royal Society Interface, written by a good colleague of mine. So this informatics approach is extremely powerful because now we have huge amounts of data. We are in the era of big data in biology. And the fact that we also have very powerful computers, I would argue from the semiconductor industry that came originally from physics. We have very, very powerful computers now. And of course, we have very powerful algorithms. This means that we can analyze this huge amount of data using AI, artificial intelligence, or machine learning, and we can pick out patterns that we'd never be able to see before. And on the left-hand side, I've just referred to the wonderful, wonderful result from AlphaFold, where they managed to use artificial intelligence to predict the folded structure of a protein. And they did a very, very good job of that. So we can see that these tools are extremely important and extremely powerful. And if we can identify these new patterns, then what's really, really, really exciting is that underlying those patterns must be some sort of physical truth, some sort of interactions that we can pull out and understand. And I've used here as an example the periodic table that started out basically as recognizing that the elements form these groups and these patterns. And then once you started to understand that, you could think about the underlying reason why. And so, and of course, in biology, the underlying reason why could be physics or it could be evolution. It doesn't matter. What matters is these informatic techniques are going to pull out new understanding that we need to then be able to rationalize. So if we can collect all of this data and we can analyze it to identify patterns, and if we can then use biophysics to understand what those patterns mean and why they exist, then we can enter the era of predictive biology. So we can go from the genotype to the phenotype. And of course, predictive biology is going to be really, really essential for personalized medicine. Because if we're going to apply something to an individual, we need to be able to make a prediction for that individual. Being able to make a prediction for the group is not going to be good enough. So we need this predictive biology for personalized medicine. So I just want to give you an example from my own work of where I found this to be helpful and exciting and powerful. It's not the best example, but it's the one I know the most about. So our collaborators who are experts in making these DNA mini circles and also in cryo-electron tomography, we worked with them to image for the first time using tomography what these tiny DNA mini circles, so they contain about 300 base pairs. So what these tiny DNA mini circles look like when they're twisted up and supercoiled. So if I just play this movie here, you can see the DNA is rotating and the superimposed structure is our model and the grid around the outside is the experimental data. So these are really, really pioneering cryo experiments because the system is so tiny. It's very, very difficult to image. So it's only when we can fit in the atomistic model from the simulation that we can get an idea of what the structure at this atomistic level might look like. So this we were able to do with our collaborators from the cryotomography, we also have collaborators who do fantastic AFM imaging with DNA. An example of this is shown at the bottom here. So we can see it's the same DNA mini circle, and we can actually see here the helical repeat. So with the AFM, we get more experimental detail. But of course, the caveat with the AFM experiments that we don't have with the tomography is that with the AFM, the DNA is forced to sit on the surface and we hit it with a big needle to do the imaging. So both of those two experimental techniques have different caveats associated with them. And of course, what we can do with the physics model, with the simulations, is we can also fit our atomistic models to the AFM data, and then we can compare whether the same model fits the cryo and the AFM, which it does, and we can then get some sort of idea whether the two sets of experimental data are compatible or not. And in this case, they really were. So so that was very nice. Just play the simulation so that you can see the DNA undergoing thermal fluctuations. And one of the things that we did pick up from the simulations, and particularly from the AFM, is that these DNA circles are supercoiled, so they're under mechanical stress. And actually these DNA circles can be so supercoiled that they form these little holes or defects or denaturation bubbles where the double-stranded DNA is actually broken, and that's what's indicated with this little red triangle here. And we can see that also in the physics models, and I've set to put a little collection of them down there at the bottom, so we can see these defects forming. And you know, the physics models, they give us some idea of where these defects will form, what the shape of the DNA should be, and it matches the experiments pretty well. So we think that certainly for a system, this is obviously a very simple system, that these models can have some predictive power. And obviously if we can move towards more complicated systems in the future, that could be very exciting. So a situation where people have done exactly that and looked at really complicated, in fact, the entire nucleo, is this particular project here, the 4D nucleo network. So the first thing to point out is that this is a big collaborative network where people have all been working together towards a common goal, and the common goal is to map the structure and dynamics of the genome, and also to look at cell populations and also single cells, because heterogeneity is everywhere in biology, and it's clearly very, very important. And so what these guys have been doing, absolutely amazing project, is they've been mapping the genome, so creating a three-dimensional contact map, and they've also then used that experimental data to build a model, a three-dimensional physics model of the nucleome. And then they, once they have that model, they then use new experiments to actually see if they can validate it or disprove it. And then this goes round in a big virtuous cycle. And the other thing that I thought was particularly exciting about this project, which I'll show you on the next page, is just how many different biophysical and genomic tools they have needed to use and also develop in order to get the information that they need to feed into the model. Because the thing is once you start to construct a model like this, you suddenly realize what's missing. And if you've got something missing, then you're stuck unless you can actually measure the missing pieces. And so that's why there's all of this experimental development that has been inspired by the creation of these three-dimensional models. So genomic technologies and imaging technologies, both of these have been developed in order to feed into this grand unified idea of the 4D nucleome, so 3 dimensions and also time. Absolutely fabulous project. So as well as the nucleome, we'd also like to, of course, be able to look at cells and tissues, and I've come up with this idea of a whole life model here. And just as a sort of thought experiment, I thought we should think about what types of information we would need to put in to this whole life model. And we realize that it needs huge amounts of expertise from many, many different disciplines, all to come together and work together. So firstly, we need to know what's there. So we need to know the abundance of things. So for that, we need informatics, we need proteomics, mass spec, all of these quantifying techniques to tell us who's there and how many of them are there. And what's really nice about this, so the website, the DigiZyme website where I've taken this picture, the really nice thing about it is if you go to that website and you can interact with these things. So you can actually put your mouse over the different molecules and it will tell you what they are. And then, of course, if you know what the molecules are, you can then get their atomistic structure from the protein database, or you can get their molecular shape from the electron microscopy database. And we can know all of these things and how they interact together. So we need to know what's there. We need to know where things are, and of course, with that we can use super resolution microscopy to try and to map things and find out where they are. We can use light microscopy. We can look at molecular shape. We're going to need to know that as well, and we can get at that with cryo-EM, cryo-ET. Ideally, for many of these objects, we won't want to have an atomistic structure, and for that, of course, we use X-ray crystallography or NMR, and we need to know what the molecular interactions are. So what binds to what interacts with what are the conformational changes, and for that we can use things like biochemical assays, calorimetry, fret-based distance measurements, etc., etc., etc. So we need all of these different tools to come together for us to be able to construct something like a whole life model. And we're doing very well at this in the biological sciences. So we're very lucky as a community that we have these amazing databases. So we have the protein database, now we have the EMDB as well for electron microscopy. We have wonderful genomics databases. We now have PDBE, and that's trying to connect together as much as possible genomics and structure. And we also have, this is very exciting, PDB-Dev. So PDB-Dev is going to be a database where we can submit what we call integrative models. So where we've used one experimental tool, another experimental tool, and combine them into a common model using probably some sort of biophysics, some sort of physical model. And what's really exciting is that if we try and bring all of this together, we're going to find out what's missing. And when we find out what's missing, this is where we want to focus our efforts, and this is where we're going to want to develop new biophysical tools to enable us to extract this unknown information. So let me just play this wonderful movie here, which is a movie of cell division. So you can see the two cells dividing here. And the thing is that the main criteria, the main determining factor about biology is that it's dynamic, it's never still. And so one of the really, really big challenges, especially for experimental biophysics, is going to be coming up with new tools and new capabilities for visualizing dynamics at multiple scales, as well as for visualizing structure. So I would say that I really like the the Allen Institute for the Cell Atlas model. These have some wonderful, use wonderful imaging tools to then try and construct three-dimensional models of human cells. This one here at the completely opposite end is one of my atomistic molecular dynamic simulations of the replication transcription complex of the of SARS-CoV-2. This example here I particularly like. So the little rail tracks that you see are labeled dynein motors walking up and down microtubules, and you can see that with imaging. And then on the right hand side next to that is a really amazing animation, actually made by a colleague of mine, Bara Malkova. And what she's done is she's imagined what types of molecular motion must be needed to generate the experimental data seen on the left. Now, of course, this is an animation, it's not a simulation. And I would say one of the challenges for theoreticians is to be able to develop mechanical models that represent this type of dynamics. So Yvette Bahar from the University of Pittsburgh came up with this wonderful quote: "Dynamics is the bridge between structure and function." And certainly in the biological sciences, we really, really need to include dynamics, or we're missing the whole point of life. So this is my view for how we go towards a particular, towards a predictive biology with help from physics in two different ways. So first of all, we need our data sets. We need large data sets, and these data sets have to be what's known technically as fair, so findable, accessible, interoperable, and reusable. And then we need methods for integrating these data sets together, so ways of combining cryo-EM and X-ray crystallography, and ways of combining super resolution microscopy with cryo-EM, etc., etc., etc. And these are probably going to be some sort of physical model, and we will need new software in order to do that. And when we can actually integrate our data sets together, we're going to find out this exciting idea of what are we missing, what don't we know, what are the important things where we need to focus our experimental efforts in order to find out unknown information. And it is very, very likely that we will need to invent new biological tools in order to do this, new biophysical tools. So for example, genome sequencing, we can sequence the genome, but we're not very good at the repetitive elements. And you know, so when we've got a whole genome map, we see that there are these bits missing, and those are the bits where we need to develop new tools so that we can access that missing information. So when we find out what information is missing, then we invent our new biophysical tools to access this information. And then we and once we have this model, this idea, this collection of different sets of data, we can start to think about what physical interactions, what physics equations do we need to represent it, and then we can get towards a predictive biology. And obviously we'll need to go round in a big circle. So we'll build our model, it will be wrong, and we'll need more data, and then we'll find out that actually there's more data missing, and we'll need a new biophysical tool, and this thing will go round and round and round and round in a circle until we get until it's probably forever, in fact, but it will get better and better and better and better each time. So I've just gone back to these two pictures from the what we understand about physics right from the very beginning, astrophysics and particle physics, and I've pulled out these two examples once more because they both involve very, very large communities of researchers working together to integrate different types of experimental information and come up with a common understanding. So these are collaborative, multidisciplinary research communities. And this is exactly what we need in the biological sciences. So it's not just the technological challenges, there are also societal ones as well. We need to be able to work together, and many different types of researchers need to be able to understand each other's language, and that's going to be more challenging in biology than it is in particle or astrophysics, because of course the complexity of biology is huge compared to anything we ever see in physics, which is why I chose to work on it, because of course it's more interesting. So on the right hand side, I have again a picture of cell division, and you can see the DNA in green, and the red shows the microtubule network that has self-assembled to enable the cell division as regulated by Aurora B. And this process of cell division involves many, many different types of proteins working together perfectly cooperatively to enable to get the cells to divide. And so given that the molecules of biology achieve this, it is unsurprising that us as researchers need to do that too in order to extract all of the mysteries of life.